In contrast to voltage-gated sodium channels, there is a wide variety of potassium channels that are expressed in neurons. We'll be devoting the next several slides to discussing the properties of the different types of voltage-gated potassium channels, and what we are showing here is just a list of the different types of channels that are expressed. First, there are delayed rectifier potassium channels. These are the channels that we've talked about before, and they're responsible for the falling phase of the action potential. A second closely related type of potassium channel is the fast inactivating or A-type potassium channels. These differ from the delayed rectifier channels in that they inactivate in contrast to the delayed rectifier channels that don't show much inactivation. And in fact, the fast inactivating potassium channels inactivate in much the same way that sodium channels inactivate. These channels are responsible for generating after hyperpolarizations that occur after action potentials. Another very important class of potassium channels are the calcium-activated potassium channels. When intracellular calcium levels increase, for instance following depolarization, when calcium channels are activated, this leads to an influx of calcium, an increase in intracellular levels, and this in turn activates these potassium channels. As we'll see, these calcium-activated potassium channels generate large after hyperpolarizations that play an important role in behavior of some neurons. And there are large and small conductance types of calcium-activated potassium channels that have somewhat different properties. Another large class of potassium channels are the inwardly rectifying potassium channels. And unlike most other voltage-gated channels, these channels gate open when cells hyperpolarize instead of depolarize. These channels contribute to the resting membrane potential of neurons and glial cells, and indeed these channels are expressed in high numbers in glial cells and are the major conductance of glial cells. And finally, there are two-pore potassium channels. These channels have a somewhat different structure than other potassium channels, and they are open, they are voltage independent, and are open at the resting membrane potential, and therefore are called leakage potassium channels, and they play an important role in generating the resting membrane potential of neurons. We begin our discussion of voltage-gated potassium channels by talking about delayed rectifier potassium channels and A-type potassium channels. These two types of potassium channels have very similar structures, and they differ primarily in that delayed rectifier potassium channels inactivate very slowly, if at all, whereas A-type channels inactivate fairly rapidly. And this is shown in these voltage clamp records down here. When we depolarize a neuron expressing delayed rectifier potassium channels, we record an outward current that doesn't decay very much over several hundred milliseconds. This contrasts with the voltage clamp record from a fast inactivating potassium channel, also called the A-type potassium channel. When we depolarize the cell, we record an outward current that then decays back within tens of milliseconds back down to zero. And the mechanism of inactivation in these A-type potassium channels is very similar to that seen in sodium channels. These A-type potassium channels have an activation gate and an inactivation gate, which is in fact the ball and chain, the same type of ball and chain seen in sodium channels. Now, because potassium channels are made up of 4 subunits, they also have 4 inactivation gates, and when any one of these swings into place to stop up the pore, the channel will inactivate. These channels inactivate at a fairly negative voltage, minus 50 millivolts, and you can take advantage of this fact to design a voltage step protocol to isolate this current and show the time course of this current in cells that are expressing other types of channels as well. And we can discuss how one would design a voltage step protocol in order to isolate a single current generated from this type of potassium channel. The A-type potassium channels are expressed in high density in dendrites, and as we will see, these channels play an important role in controlling and modifying the electrical properties of dendrites. Calcium-activated potassium channels represent another very important class of voltage-gated potassium channels. And there are two basic types of these channels. There are BK channels and SK channels. The BK channels are so named because they have a high unitary conductance of about 200 to 400 picosiemens. Therefore, they're called big potassium channels or maxi potassium channels. These channels are directly gated by intracellular calcium. The structure of the BK calcium-activated potassium channel is shown here, and on the C-terminus there is a binding site for calcium, and when calcium binds to this site, the open probability of the channel increases. This channel is also voltage sensitive, so both depolarizations and increases in intracellular calcium lead to an increase in the open probability of this channel. This channel can generate a fast hyperpolarization after hyperpolarization, the AHP, and this channel is blocked by tetraethylammonium, TEA, as well as charybditoxin. A second type of calcium-activated potassium channel is the SK channel, so named because it has a small unitary conductance of about 2 to 20 picosiemens. In contrast to the BK channel, the SK channel is modulated indirectly by calcium. When calcium binds to calmodulin, this leads to a binding of the activated calmodulin to sites, again, on the C-terminus. And in contrast to the BK channel, the SK channel is voltage insensitive. So it's only modulated by calcium via calmodulin. This channel can also generate after hyperpolarization, and it is blocked by apamine, which is a toxin that is isolated from bee venom. The diagram in the upper right shows the current voltage relation recorded from cells expressing BK channels. And this diagram illustrates that the BK channel is sensitive and controlled both by voltage and by calcium. When calcium within the neuron is at a low level, 0.1 micromolar or 100 nanomolar, we see that the open probability of the channel is very low, never increases more than to about 5%, even when we depolarize the cell to plus 50 millivolts. However, if we increase intracellular calcium levels to 1 micromolar, 10 micromolar, 100 micromolar, we see that the IV curve shifts to the left, increasing the open probability. So for instance, when the intracellular calcium concentration is 10 micromolar, we see that although the channel is closed at negative voltages, as we depolarize, the open probability of the channel increases. So this channel is modulated both by voltage and by intracellular calcium levels. The diagram in the lower right shows a voltage recording from a cell expressing SK channels, the small conductance calcium activated potassium channels. And note that this is a very slow time course, and what we're showing here is a depolarization which triggers a number of action potentials. And following this, which actually leads to a calcium influx into the cell, there is a very strong after hyperpolarization, And this after hyperpolarization can be blocked by adding apamine, which blocks the SK channels, and in the presence of apamine, the after hyperpolarization is much smaller. So this component of the after hyperpolarization was generated by SK channels. The remaining component was perhaps generated by PK channels.